Hello everyone. In this video, let us look into the types of fermentation. Microbiologists use the term fermentation in two different contexts. First, in metabolism, fermentation refers to energy generating process where organic compounds act as both electron donor and electron acceptor. Whereas if you look into the industrial microbiology, the term refers to the growth of microorganisms in large quantities within the vessels that are referred to as fermenter or a bioreactor. Now, the fermentation process can be carried out in different conditions. If they are carried out under sterile conditions, then we call that as an aseptic fermentation. If we do not carry out fermentation in a sterile condition, then we call it as aseptic fermentation. We also have aerobic and anaerobic fermentation depending upon the microorganisms which are used. If we use aerobic organisms, then we call it as aerobic fermentation. If we use anaerobic microorganisms for the fermentation process, then we call it as anaerobic fermentation. Then based on the running system and the regulation, we have two types of fermentation, that is batch and continuous fermentation. And based on the growth pattern, we have submerged fermentation and surface fermentation. Also, if we use a solid media for the fermentation, then we have another type of fermentation called as solid state fermentation. Now in this video, we'll be concentrating only on aerobic and anaerobic fermentation. In my further videos, we will look into the batch or continuous and submerged or surface fermentation. So first, if we talk about the septic fermentation or aseptic fermentation, as I told you, if we are carrying out the fermentation under sterile conditions, then we call it as an aseptic fermentation. So aseptic fermentation includes animal and plant cell cultures, production of alkaloids, amino acids, or production of acetone, butanol, ethanol. All of these are examples of aseptic fermentation. Now, if we look into the septic fermentation, this we do not carry out under sterile conditions. Example could be the production of alcoholic beverages and silage production, anaerobic wastewater treatment, then ripening of some cheese, mushroom production. They all come under septic fermentation. Moving on into aerobic fermentation. Depending upon the oxygen requirements for respiration, we all know that microorganisms are categorized into aerobic and anaerobic, although there are some intermediate type present like facultative aerobes and anaerobes. Majority of the microorganisms that we use industrially are aerobic organisms. They include filamentous fungi, yeast, actinomycetes, and some bacteria. All the animal and plant cell cultures also respire aerobically. The only anaerobic microorganisms that we use in industry is Clostridia, which is a bacteria. Now, in aerobic fermentation, in the beginning of the fermentation process, small amount of oxygen will be dissolved in the medium which will be available to the microorganism. However, very soon it is exhausted, creating an anaerobic condition. Therefore, an aerobic fermentation system needs a continuous supply of either air or pure oxygen under aseptic condition, that is under sterile condition. We should be supplying air or a pure oxygen. The method for provision of a culture with the supply of air varies with the scale of the process. At what scale you're going to do the process, with that scale the method for the supply of air will vary. Industrial scale fermentations are normally carried out in stirred aerated fermenter, wherein there are impellers which keep stirring the mixture, which helps in the continuous mixing of oxygen, which are supplied by a device known as sparger. Now, the sparger is the device which will be fitted at the base of this stirred aerated fermenter, which will supply sterile air into the fermenter. So, the aerobic fermentations are usually carried out in stirred aerated fermenters. Some fermenters are designed to supply the adequate oxygen transfer without agitation. Such fermenters are bubble cap fermenters and airlift fermenters. So, in aerobic fermentation, we continuously supply oxygen through spargers, the device which helps to supply the air into the fermenter. Moving on into anaerobic fermentation. Anaerobic fermentation takes place in a closed airtight fermentation system The anaerobic conditions here are created by different methods. As I told you, initially when we put for fermentation, a small amount of oxygen will be present in an empty fermenter tank and in the media. So therefore, we usually gas the fermenter tank with the carbon dioxide after sterilization to remove all the oxygen that is present in the fermenter and in the media. Therefore, an anaerobic condition is created for the anaerobic microorganisms to grow. The examples of anaerobic fermentation are acetone fermentation, butanol fermentation, ethanol fermentation, glycerol fermentation, and some toxins production. And aerobic fermentation includes mushroom production, steroid biotransformation, some vaccines production, vitamins production. They all include aerobic fermentation. So now in my next video, we will look into batch and continuous fermentation. Thank you.